So today's notes are going to be on carbon and carbon-based life. Making those connections again to our big ideas. We have big idea 1, evolution, and we're going to see that all life forms on Earth share similar or the same biochemical similarities. They have the same macromolecules that make up life. Okay. And then big idea 4, life forms include large, complex molecules with common building blocks. Those complex molecules make interactions with each other, therefore interactions. If you've ever heard it said that we're carbon-based life forms, that's because we all have carbon, all living things have carbon as a base for a lot of their molecules. And as a matter of fact, an organic compound is usually one containing carbon. There are 4 classes of larger molecules, that's the macro, that we're going to talk about, that are present in all life, or in all living things that we currently know of. And those are proteins, carbohydrates, lipids, and nucleic acids. 3 of those we can get from our diets, 1 we cannot. So let's talk a little bit about carbon. Carbon is an element on the periodic table. It has 6 electrons, okay? 4 of those electrons are valence electrons, so 2 in its first shell, 4 in the second shell. That second shell wants to have 8 to make it complete. So carbon is a really good sharer, loves to share electrons with other atoms. And when we have that sharing, remember, those are called covalent bonds. Now sometimes carbon will share those in a single bond, so 1 pair of electrons, or it'll share it in a double bond, 2 pairs of electrons. So for instance, if carbon is sharing with hydrogen, it's only sharing 1 pair of electrons, but if it's sharing with another carbon, it's sharing 2 pairs to make that complete. Each carbon atom acts as an intersection point and can branch off in up to 4 directions. Okay. So sometimes you'll see, as in methane, which is CH4, the carbon will be in the very center, and then you'll have a hydrogen, 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 and hydrogen bonded to it. Sometimes you might see, as in ethylene, we have the carbon double bonded to another carbon, so this is C2H4, and then we'll have an H, an H, an H, and an H in single covalent bonds. Carbon has some very frequent partners, which include hydrogen, oxygen, and nitrogen. The carbon that forms the backbone of many of the structures, compounds that we will talk about, we'll call that a carbon chain. Some of those chains are straight, some of them are branched, and some of them are in closed rings. Some of them will have single bonds and some of them will have double bonds. We're going to talk about hydrocarbons because they're important to life, especially in relation to fats. Hydrocarbons only have carbon and hydrogen, thus the hydrocarbon. We're going to, like I said, we're going to find these in fats. Fats have long hydrocarbon tails. They are really good at releasing large amounts of energy and storing energy, and they don't dissolve well in water, which makes them a really good storage. So it's not just plain hydrogen or oxygen that will attach to the carbon skeleton. Sometimes we'll have, we'll have chemical groups that are attached to these carbon skeletons. The number and arrangement of each of these carbon or chemical groups, gives the molecule its unique properties. Okay, so some chemical groups are going to function by affecting the shape of the molecule, and some chemical groups will affect the function by being directly involved in chemical reactions. And we're going to call these a functional group. Functional groups will come out again when we talk in our other big ideas, especially big idea 2 and big idea 4. Page 35 of your textbook has a table of functional groups that will be formed on those organic compounds. That list on table 35, though, only includes 5 of the main functional groups: hydroxyls, carbonyls, carboxyls, aminos, phosphates, but also leaves off the sulfhydryls and the methyls. That's because methyl is not reactive, but it does serve as a tag on biological molecules to help them recognize each other. Frequently, there'll be a tag on a cell that needs to be destroyed, or a molecule that needs to make it through a membrane, or a molecule that needs something else attached to it. So that's what they mean by a tag. All of the 7 groups above, except the sulfhydryls, are hydrophilic, which, again, remember, means they like water. Okay. And they actually will help the organic compound to be able to be soluble in water. And water, from last chapter, is the solvent of life. In regular biology classes, you may have heard of ATP as the cell's energy. That's kind of how we learn it, the currency of the cell. Well, ATP is adenosine triphosphate, so it has an adenosine with 3 phosphate groups. Okay, so that was that the adenosine is that carbon skeleton, and then the 3 phosphate groups, the functional groups that are around it, are going to store the energy. When those are ripped off, then we get energy release. When they're put together, so when we have adenosine diphosphate and we add another phosphate group, we're putting energy into that molecule. Our main focus, however, is going to be on 3 macromolecules. And I put the title here as Monomers and Polymers. Oh my, because we're going to talk about what is a monomer, what is a polymer, and how this all works, and DNA, and carbohydrates, and proteins, and a little bit about lipids too. Okay. So polymers are long molecules made up of similar or identical building blocks, and those building blocks we're going to call monomers. They are all linked together by covalent bonds. So one example I have here for you is if each individual train car is a monomer, so they're similar or identical, right? They can be linked together and form the whole train. Another example, if you don't like the train example, you can think of Legos. When you have a whole bunch of Legos, those are all the monomers, each Lego piece. And then when you put them together to make a structure, the polymer, or you're making a house or a car or whatever you build with Legos, then you get small pieces making up a big piece. Okay, so a couple things about polymers. They can be assembled by a dehydration reaction, which means we're going to lose water off of that carbon skeleton with the functional groups attached. So we're going to actually lose water, and then we're going to put them together. Okay. Or we can break down a polymer by using enzymes that are going to speed up hydrolysis, which is using water to break them apart. So water, again, solvent of life, involved in lots of different things. We're either going to lose water to put a polymer together, or we're going to use water to break it apart. So an example here for life is in our digestive system. you eat food, it goes down, your stomach breaks it up a little bit into smaller chunks, but then it'll go into your intestines, and your intestines send in enzymes to attack the polymer, which your food, okay, your big chunk of food, and by adding water, hydrolysis is going to occur, and we're going to break apart that big structure, that big food, into the monomers, which are usually glucose that we're going to be consuming. Okay, so this is why it's important to drink lots of water. In each cell, each of our cells, we have thousands of different macromolecules. That should not be an apostrophe S, sorry. Okay, these are constructed by only 40 to 50 common monomers. Don't worry, you don't need to learn all 1,000 macromolecules, and you definitely don't even need to learn the 40 to 50 common monomers. We're going to focus on glucose a lot and a little bit of sucrose and fructose and a few others, but not all 40 to 50. Okay, but what we can think about is, you say, wow, how do we get thousands of different macromolecules from only 40 to 50 common monomers? Think about the number of words that we can construct from our 26-letter alphabet, depending on how many of those letters we use and the arrangement of those letters. And so the arrangement becomes key. Differences between siblings are pretty vast. Differences between individuals are extensive, and differences between species are even more so. So let's get into the carbohydrates. Carbohydrates are sugars and polymers of sugars, so simple sugars and more complex sugars. Usually they're named ending in OSE, okay, and sometimes by the amount of carbon that they have. So for instance, a pentose, we have the pent and the ose, so the ose is the sugar, and then the pent is the 5, so 5 carbon sugar. Hexose would be 6 carbon sugars. Simple carbohydrates, we're going to call those monosaccharides. Mono usually means one, so we're referring to something that's simple, okay? Or we can also call them simple sugars. Saccharide is a sugar. They're usually going to be in multiples of this CH2O, so carbon with a water. So let's look at glucose. We have C6H12O6, okay? So CH2O, combination of that. And fructose actually has the same chemical formula. They do have a carbonyl group and multiple hydroxyl groups attached to them. And remember, you can go back to page 35 if you are questioning what those look like. And the cells can extract energy from glucose during respiration by breaking down molecules. So this is why it's important to us. We need to be able to get energy from the food we eat, so we have to break it down, even the simple sugars. Then we have disaccharides, which are double sugars. This would be like sucrose or table sugar. Another type of disaccharide is lactose or milk sugar. Many people have lactose intolerance. It's actually less prevalent in people of European descent than it is in people of other descent, okay? But there still are about 17 to 20% of Europeans that have lactose issues. They cannot digest lactose. After a certain age, somewhere around 2, the amount of lactase, which is an enzyme that you use to digest lactose, starts to decline. In European descent, that's not as prevalent as in like African descent. That is almost completely gone after the age of 2. It just keeps getting worse and worse and worse. Then we have polysaccharides, hundreds to thousands of monosaccharides put together. This would be like making a really big, huge Lego structure, or If we want to use the train example, those trains that you get stuck at that seem like they're like 6 miles long, that would be like a polysaccharide. And they're going to serve as storage materials or building materials for our cells. So let's talk about storage and how those polysaccharides play a role. So plants and animals both are going to store sugar for later use. Plants store it differently than us. Animals store sugars for later use as something called glycogen, which is a branched glucose, and it's stored in the liver and in muscle. Okay. But in humans, this can only last for about a day. So it doesn't last very long. It's not a very stable, stick-around storage structure. We replenish our stores of glycogen as we eat. So you eat something, it gets broken down, it gets stored as this polysaccharide called glycogen, and you use that up throughout the day until you eat again, and then you replenish your stores of glycogen. Now, plants, on the other hand, they store starch, which are multiple glucoses put together. And this can be long-term storage because they have grains and tubers and other parts, fruits of their plants that they can store them in, and that doesn't seem odd. But to us, that would be odd if, you just stored everything in your leg. Then your leg would be really huge and nothing else, right? Okay. So then we have those were the storage. Now we have the structural. Structural are two different types, cellulose and chitin. Okay, cellulose we're going to find in plants. It's in their plant walls. That's actually one of the properties that makes an organism a plant. If you took botany, you might remember that they have cell walls with cellulose. Cellulose is the most abundant compound on Earth because most plants make it. There really aren't very many plants that don't have cellulose. Cellulose is straight and never branched, which is going to tell you a little bit about its function and its structure. It's like fibers. Okay. Now, the problem with cellulose is that few organisms actually have the enzymes capable of breaking apart cellulose and being able to digest it. This is good for us. If you've ever heard, you need your fiber every day. It's very good to have lots of fiber. Well, that's basically the cellulose that they're encouraging you to get. Cellulose is insoluble. We cannot break it apart. It does not go into solution with water. So we call it an insoluble fiber, and it passes through us, which helps us go to the bathroom, right? So that's why we need lots of it. Helps keep our system flowing and clean. Some microorganisms actually do have the enzymes that are able to digest cellulose, and those are bacteria and protists. Now, you're going to find a lot of those bacteria in the gut of a cow who's able to digest the cellulose because of those bacteria. Another place you're going to find those are protists will have these enzymes, and they'll live in the gut of termites, which eat wood. Okay, so the termite itself can't digest the wood, but it has a symbiosis with the protist who can break down the wood for the termite. Okay. And then we also have the structural polysaccharide called chitin, and this is used to build exoskeletons in arthropods, and also we'll find it in fungi, but not as hard and not as structured in the fungi, because you don't like bite a mushroom and it like doesn't crunch in your mouth. That would be disturbing. Okay. Now, chitin is similar to cellulose, but it does include nitrogen, whereas cellulose does not. Now we're on to another group of macromolecules, the lipids. These are large molecules. They do not consider them true polymers. Okay. They are mostly hydrocarbons, and they have nonpolar regions. So fats are nonpolar. And they're going to include fats, phospholipids, and steroids, all of which are very important to human life. And we all know fats and water do not mix, or oil and water do not mix. So let's talk about those fats first. Fats are large molecules that are put together from smaller molecules by dehydration. That process of putting two smaller molecules together is going to store energy for us. They have glycerol. Glycerol is an alcohol, and the carbons all have hydroxyl groups on them. And fatty acids, which are chains of 16 to 18 carbons with carboxyl groups on them. Again, refer back to that chart on page 35 if you want a better visual of what I'm talking about. Okay, then the 3 fatty acids can be joined to that glycerol, and we're going to call this a triglyceride. So 3 fatty acids, the tri and the glycerol, so the triglyceride. There are 2 main types of triglycerides. Those are the saturated fats and the unsaturated fats. The saturated fats have no double bonds between the carbons, and they are full of hydrogen. These are going to be most animal fats, and they are solid at room temperature. Okay, so if you think about what that does for us as we're eating that, and we have to break that apart. Whereas the unsaturated fats have 1 or more double bonds between them, and these are going to be the plant fats. Those bonds are going to hold things a little bit further spaced apart, which is going to keep them in the liquid state. So you're going to want to get more unsaturated fats in your diet than saturated. One thing that you want to be aware of when you're looking at food labels is stay away from things that are called hydrogenated vegetable oils. This means they're actually putting they're actually saturating that fat, that plant fat, with hydrogens and causing it to become a saturated fat. Not good. Another type of lipid that we should talk about are phospholipids because they make up the cell membrane. Got a little picture down here of the cell membrane. Okay. So the cell membrane is going to have 2 fatty acids, a glycerol, and a phosphate group. That's why we refer to it as the phospholipid. So here are the phosphates up here, and then here facing towards each other are those hydrophobic tails, or the fatty acids with the glycerol. So when we get these phospholipids together, when we put them in a solution of water, what's going to happen is they actually spontaneously form these membranes because those tails, those lipid tails, do not like water, and the phosphates are okay with water. They're hydrophilic, they like water. So they will actually spontaneously just turn their tails into each other to kind of like protect them, kind of like if something is trying to attack a baby rhino, all of the other rhinos will form a huge circle around it. So it's like a protective circle. So we could say that cell membranes can actually spontaneously form just based on their structure, but they won't form as a single layer. You'll only see them as that double layer to protect those fatty acids in the middle. Okay, and then finally we have steroids as our last group of lipids. These have carbon skeletons of 4 carbon rings fused together, and there's going to be different steroids, and they differ by the chemical groups that are attached to them, those things on page 35 of your textbook. Okay, Cholesterol is one of the most important steroids that we have in humans and in animals. It's important in our cell membranes. It also makes our sex hormones. Okay, so that's the start of the sex hormone is the cholesterol with more chemical groups attached to it. They are made in the liver, and we can also get them from our diet. Low levels, normal levels, great. Too high of levels are not good. We've all heard, oh, your cholesterol is too high. That means you're taking in too much of it from your diet. Your body will put together what it needs and then supplement from your diet. But if you get too much in from your diet, then it can cause a buildup or in your arteries and can actually cause a heart attack. Okay. Now we're on to another group of macromolecules called proteins. Proteins are huge in cells. They make up about 50% of the dry mass of cells. If you remember from our last notes, we said that cells were 70 to 90% water. So the stuff that isn't water, 50% of that is actually proteins. They are instrumental in almost everything an organism does. And you might have learned about that in your regular biology class when you learned about cells, and protein was one of the things that you learned about, and it was one of the main functions or products that the cell is going to produce. So proteins make up enzymes. They are part of our defense systems, our storage systems, our transport systems, communication within the cells, movement, and also they play a role in support. We have tens of thousands of proteins, and each protein is going to have a unique three-dimensional shape. We'll talk about how that happens a little bit. Proteins are polymers, larger molecules made up of smaller monomers called amino acids. And when we get those amino acids together, we're going to call it a polypeptide because of the bond. We'll get to that in a minute. Enzymes are mainly proteins that are going to speed up reactions. So very important for us. Without enzymes, a reaction might take a really, really long time to happen, but with that enzyme, we're going to speed up that chemical reaction and make it occur at a faster rate. Okay. So let's talk about those components of proteins. The amino acids. If you remember from your biology classes, you went through protein synthesis. So the DNA made RNA, messenger RNA, and then messenger RNA was the message or the code, and it went to the tRNA and the rRNA, the ribosomal and the... So sorry if I repeat something. My dog actually just got sick, so I just paused the recording. I just had to clean that up. Okay. So when we do the protein synthesis, we said that the DNA codes for a specific amino acid that the RNA goes and gets, and they assemble these together. And there are 20 amino acids. Thank you, AP, for actually making a change. You used to have to remember all 20 of those, so yay, shout out. We don't have to remember all 20 amino acids. But an amino acid is an organic molecule, so it has carbon, and they all have amino and carboxyl groups attached to them. Where they're attached to them and how many other functional groups they have on them will determine what they actually do. They have and that functional group we are going to call the R group or the side chain. And I do have a picture of that right down here. Okay, so the R group or the side chain is going to be different with each amino acid, making it the 20 different ones and giving it unique characteristics. Okay, so when we put together a polypeptide, we're actually putting together many peptide bonds. So when we have two amino acids, we're going to position them so that the carboxyl groups are next to the amino groups, and then dehydration is going to happen. Okay, so we'll see if I can grab these and move these pictures. So we've got the carboxyl group and the amino group are going to attach to each other. And actually right here we can see the H2 and O. So we're actually going to make a water, and then the water will be released, and then these will be attached. Okay. This is called a peptide bond, and it's going to be repeated over and over and over again, and we keep getting more amino acids strung together until we get to that stop codon, or that place where the messenger RNA has the message that says, nope, we're done, stop right here. This is done with this protein. Okay? And remember that each one is going to have those functional side groups or R groups. So we have many, many different arrangements that can come from just those 20 amino acids. As far as then how does that make a protein, because it's not just one big long strand of it. Proteins are, we said there's thousands of them, and their activities are determined by their structure, which has different parts to their structure. First thing, of course, is sequence that comes from the DNA, which amino acids follow which ones, okay? And then there's folding, twisting, coiling, or one or more polypeptides together. So chains of these amino acids put together will make a protein. Okay. Many proteins have a round, sort of spherical shape, but some also have a long, kind of fibrous shape to them. Their function depends on their ability to bind to other molecules. So when we go back to the endorphin example that we talked about, the endorphins are a protein that's going to be able to bind to a receptor, so it's going to fit like a lock and a key. Well, the opiates have that same lock, or that same key that fits that lock, which is why they're going to be able to do the same thing for us. Not that I'm encouraging you to do opiates. You should get your natural endorphins by, just being happy with life. Okay, so now let's talk about those levels of proteins, or how that protein comes together. And we talked about, like, sequence was first, okay, but then there's different levels, and those are primary, secondary, and tertiary levels. A quaternary level can also happen when we have multiple polypeptides, so chains of amino acids again. Okay, so primary structure of a protein is a linear chain. We're just putting it together, right? Okay, then secondary, we've got all of the polypeptide is to put together, and then it's going to start to coil and fold on itself based on hydrogen bonds. So based on those functional groups, those R groups that they have attached to them, the side chains, okay? It's going to determine how it twists and folds because of hydrogen bonds. And then we have the tertiary level, which will actually be more interactions between those side chains depending on what they are. Okay. And then finally we can have a quaternary level of a protein, or a folded protein, if there's interactions between multiple chains of amino acids. So again, we had first, which is just the chain. Second is going to coil because of hydrogen bonds. Third, we're going to have interactions between those side chains and bonding depending on what those side groups are. And then the quaternary would be multiple polypeptides together. So sometimes a protein can be made up of many different chains of amino acids all put together. Sorry, an example of that quaternary would be something that we know as collagen. Okay. So sometimes when something goes wrong with the amino acid sequence or with the protein itself, we can have diseases like sickle cell anemia or other problems. So sickle cell anemia is an inherited blood disorder, and we know it's caused by a change in one amino acid at that primary level, that linear level. So one change causes sickle cell anemia. And what happens is because there's that one change, it's going to cause a change in all the other levels of the protein, and the blood cell is actually going to deform because of that. And then it can't accept the oxygen like it's supposed to, and then people get very weak and sick from that. There can also be misfoldings of proteins, and sometimes that happens to different individuals. It doesn't necessarily have to be super late in life, but usually in the mid to later life areas, we get a misfolding of proteins, and we can get Alzheimer's, Parkinson's, mad cow disease. Okay. Mad cow disease is an accumulation of those. And then sometimes, I don't know why I just drew on the screen there, sorry guys. Sometimes we can get a denaturing of proteins. We see this a lot if you ever hard boil eggs. This is caused by changes in pH, the changes in salt, or temperature, as in the egg whites. What's happening when we put the heat on the egg, we're actually destroying weak bonds. The proteins are going to unravel and lose shape, and then we get that change of color, change of structure. Finally, we have our last group, which are the nucleic acids. Nucleic acids are DNA and RNA. They provide directions for the cell. DNA, remember, stays in the nucleus unless, of course, it's a bacteria where it's going to just be in a circular structure that we'll call plasmid. But we get to learn about those a little bit later in this class. Okay. DNA, what's really cool about DNA is that it is a set of instructions, but it has its own instructions for replicating it within it. So to kind of think about that, that would be like if you were reading an instruction book, and in the instruction book it told you how to make more copies of the instruction book. I don't know, I think that's kind of cool. Okay, so DNA makes the RNA, or directs RNA to be made. Messenger RNA we just talked about, goes out and finds the rRNA, the ribosome, and then we're going to put those polypeptide chains together, the amino acids. Okay, so but we have to talk about what actually is the nucleic acid, and that is the DNA. They are polymers of monomers, which we're going to call nucleotides, and I'm sure you all remember from all your biology classes going over the parts of a nucleotide, so we're going to go over those again and then just talk about what it is. So DNA is not involved in running cell activities, but it is the inherited material. So sometimes, maybe throughout middle school or early high school, we kind of said the nucleus was the control center. And in a way, yes, because it holds the directions, but in a way we've got to kind of amend our thinking on this, and that when I think of control center, I tend to think of the place that runs everything. And DNA really doesn't do that. It just is the container that holds the directions. So the next time you get a new cell phone, or if you think about when you had a new cell phone and you got that little, inside the box is the little tray, and inside the tray is the book of instructions that unfortunately, we usually don't look at. For DNA, we're actually looking at it all the time to find out how our bodies function. Okay, so it's more like just that tray that holds the instructions, the nucleuses. It doesn't really direct or control what's happening. Okay, I digress, sorry. So each chromosome that we have, and humans have 46 chromosomes, 23 from mom, 23 from dad. It contains one super long DNA molecule with hundreds of genes or sections. So we've got this super long DNA molecule, and we can break that apart into little sections or that we can call genes. So if we have, and this is a very short example, but if we had like A, T, G, C, T, A, that would be a gene. Okay, and then with the next sequence of letters, we could start another gene. So each DNA molecule is made up of several hundred genes, and then each chromosome is one whole DNA molecule. DNA, we said, directs the making of messenger RNA, which then directs the making of the proteins by amino acids together. So we have the nucleic acids, which are the monomers of DNA and RNA, and they have three parts. They have a nitrogen-containing base, so we've got either one or two rings that have both nitrogen and carbon, because they're organic molecules. The pyrimidines have one ring of 6 carbons and then nitrogen, and those are going to be cytosine or C, thymine or T, or uracil, U, and RNA, because remember in DNA we have thymine, but in RNA we have uracil because of how everything fits together. And then the purines are going to be one ring with 6 carbons and one ring of 5 carbons with nitrogen in them, and those are going to be adenine and guanine. Okay. The sugar that's part of the nucleotide is deoxyribose or ribose. It's a 5-carbon sugar. And then finally there's going to be a phosphate group attached, one of those functional groups, remember? Okay, so let's just review that real quick. Nucleotides have 3 parts: the nitrogen base, A, T, G, C, or U, and then a 5-carbon sugar, deoxyribose or ribose, and then a phosphate group. Okay. And then just some quick differences between DNA and RNA. DNA has 2 polynucleotides or strands, so we've got, one strand goes up, the other strand goes down, and they come together with hydrogen bonds in the middle, and it's going to form a double helix. So when you talked about DNA replication in biology, you talked about the enzyme coming in helicase and breaking those hydrogen bonds in the middle between the 2 nitrogen bases. So that's where those hydrogen bonds come in. They have, like I just said, this anti-parallel arrangement. So, traffic's going one way on one side and the other way on the other side. And we're going to call those the 5' to 3' side, or the 3' to 5' side, depending on the sugar and what's hanging off of it. Okay, we'll get into that a little bit more. On the outsides, we have alternating phosphates and sugars that are all bonded together. And again, those nitrogen bases on the inside together with hydrogen bonds. Adenine on one side will always pair with thymine on the other side through the hydrogen bond. Guanine on one side will always pair with cytosine on the other. Now, RNA only has the one strand, so there's no pairing going on there, and it's going to use uracil instead of thymine. Okay. So please, again, remember to ask questions. Anything that you're unsure of, don't understand, want to go over further in class, you can all take a look in your book, see if there's anything that can be answered through there, and please ask me lots of questions when we get to class. Okay, I will see you guys later. Bye.